plant growth, where we discussed about two types of growth in plants, that is arithmetic growth, which is linear, and second was geometric growth, which is exponential. Right? So it gives the linear graph, while this one was the exponential graph. And then we discuss about various plant growth regulators that actually plays a role in the development. So these plant growth regulators are the signaling molecules in the plant. They are the signaling molecules. They are the signaling molecules in the plant which are secreted in very low concentration, and they may act locally as well, unlike animal hormones. So these are plant growth regulators, and we divided plant growth regulators into two categories. First one was the growth activator, growth promoting. The second one was growth inhibiting, that inhibits the growth. So in this category, we put auxin, cytokinin, and gibberellic, that is gibberellic acid. And in this category, we put abscisic acid ABA, and ethylene at some point can be considered in both the categories. Also within these growth promoting and growth inhibiting, there are certain more plant growth regulators which are being identified. So we will learn about the major properties of these 5 plant growth regulators which are mentioned in NCERT, and we will also have a brief discussion on the new plant growth regulators which are identified. So we discussed that auxin. So firstly we will start with auxin. Auxin, as we know, was firstly discovered by Darwin and his son Francis, who observed photoperiodism in the plant. Phototropism in the plants. So sorry, who observed phototropism in the plants. So in this, what was happening is, there was a bending of shoot towards the light. So this was observed by Darwin and Francis, and later on the molecule which was responsible for this phototropism was identified to be auxin. So auxin, the term auxin is derived from a Greek word that means to grow, and it is any chemical substance that promotes the elongation of coleoptiles. Although auxins have multiple functions in flowering plants, but the main function which is related to the auxin is the elongation of coleoptiles. That is the tip of the shoot. So auxins can be both natural. Auxins can be both natural as well as there are synthetic auxins as well, which are being synthesized. So the major natural auxin in plants is indole acetic acid, IAA. This is important because most of the times the term auxin is not used. Instead, IAA is used in the questions. So the term indole acetic acid is used for the natural auxin, which is identified in plants. Although several other compounds, including some synthetic ones, also have this auxin activity, that is, also have this elongation of coleoptile activity. But the natural one is indole acetic acid. So IA was the first plant hormone which was to be discovered, and it was first isolated from human urine. And it was first isolated by A.W. Wien. So auxins like AAA, that is indole acetic acid, which is a natural one, and indole butyric acid, which is also a natural auxin, are isolated from plants. However, beside this, we have some synthetic auxins as well, like NAA, which is naphthalene acetic acid, and 2,4-D, which we also know is used as a herbicide. So this 2,4-D is a 2,4-dichlorophenoxyacetic acid. So they are the synthesized auxins. So natural ones example include indole acetic acid and indole butyric acid, IAA, IBA, and the synthetic ones include NAA, that is the naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic acid. Biosynthesis of auxin occurs by the tryptophan amino acid in the presence of zinc ion. Therefore, the presence of tryptophan is important. In the presence of zinc ion, the biosynthesis of auxin takes place, and it occurs at both shoot and root tips, although shoot is the predominant site. So it is produced predominantly in shoot tips and is transported from cell to cell down the stem. So down the stem, suppose this is the direction of stem. So this is the top. This is the top, and this is the bottom. So auxin will be transferred from this to this, cell to cell, down the stem. Now, the another question that arises, if I changes the orientation of this stem, then what will happen? So will it be like this or like this? So it will not be like this. If I changes the orientation, again the auxin will move against the gravity. So auxin is not affected by gravity, but is actually affected by the concentration and moves from coleoptile region to the downward cells. To downward cells. So this is important. This is a unidirectional movement, and it moves only from tip to base, not in the reverse direction. So it will not move in the reverse direction. It will only move from tip to base. And this unidirectional transport of auxin is called the polar transport. So there is a polarity in the movement. For example, if I called it as positive end, I called it as negative end, it will always move from positive end to negative end. So there is always a polarity. So it will always move like this. So the direction may change, but if I reverses the direction of the stem, but that does not mean that the polarity has changed. Polarity remains the same, and this unidirectional transport of auxins is called the polar transport. So this is important, that the transportation of auxin is not affected by the presence of the gravity. So it is not under the effect of gravity, but it is actually a unidirectional transport that happens from cells that are present on the tip to the base. The cells which are present on the in the tip region, the auxin moves downwards to the base region. Right? One of the very important effects. So now we will learn about the physiological effects of auxin. So one of the very important effect of the auxin is the apical dominance. So this is one of the very important physiological effect of auxin. What actually is apical dominance? So it is a phenomenon in which the apical bud dominates the growth of the lateral bud. So there are two types of buds. The one which is present on the apex, which is the apical bud, and then we have lateral buds. So apical dominance, as the name suggests, when the apical bud dominates the lateral bud. So the growth of the lateral bud is dominated by the apical bud, and this phenomena is called as the apical dominance. So this is important phenomena which is very important in terms of auxin. So it is one of the very important physiological effect of auxin. So you can see here that apical dominance in plants has been shown here, in which if I consider figure A, so in this the apical bud is intact. So intact apical bud is there. And in plant B, we have removed the apical bud. So you can see here that there is a growth of the lateral buds after the decapitation, that is after the removal. You can see here that there is a result. What we will see is there is a result in the growth of the lateral buds. But when there is an intact apical bud, lateral buds growth stop. Lateral bud will not grow, while in this case, lateral bud will grow after decapitation. So what is decapitation? Decapitation is the process of removal of short shoot tips. That is the region where why we are doing this? This is the region where auxin is present, right? So if I remove this region, the effect of apical dominance subsides and the lateral buds grow. So the growth of the lateral buds is dominated by the growth of the apical buds, and upon the removal of the shoot tips, that is upon decapitation, growth of the lateral buds can be seen. So it is widely applied in tea plantations, hedge making, etc., where the farmers on the daily basis they remove apical bud. They remove apical bud to allow the growth of lateral buds. Right? So this is important physiological effect of auxin, where if the apical bud is present, then lateral growth stops, and if apical bud is removed, lateral bud will grow. The second is the cell division and cell enlargement. So mainly it helps in the cell elongation. Cell division is the main function of the cytokinins that we will see. So cytokinins plays an important role in cell division, but usually the auxin plays an important role in cell elongation. It helps in cell division because once the cell is elongated, it prepared itself for the cell division, and auxin controls the xylem differentiation. So these are the important points that we need to remember, that it leads to cell elongation that helps in the cell division, and it controls the xylem differentiation. Next is root initiation. So rooting on stem cuttings widely used in plant propagation is promoted by IBA and NAA. That is, it is promoted by applying auxins. So IBA, which is a natural one, NAA, naphthalene acetic acid, which is a synthetic one. So when when there are low cytokinin concentration, this we will see later also, when there is low cytokinin concentration and high auxin concentration, root differentiation happens. So it is not the effect of only one hormone. We know that there are multiple hormones that can act together in order to carry out a particular function. So the fourth is prevention of abscission. Abscission means the falling of leaves or older parts of the plants. So IAA prevent premature abscission. It prevents premature falling of leaves or plant organs and promotes the abscission of older mature leaves and fruits. So this is important. Anything that is helping the growth. We do not want young parts to fall, and we want older parts to fall so that nutrients can be used by the young ones to grow. Therefore, anything which is mostly related to the growth, that is helping in the growth of the plant, is the function of plant growth regulators. The prevention of abscission is one of them, where IAA or NAA can prevent the premature abscission, that is the premature falling of the plant organs, and it promotes the abscission of the old mature leaves or fruits. Flower initiation. So auxin promotes flowering in pineapple and lychee plants. Therefore, it is also very important from the commercial point of view. Parthenocarpy, that is seedless fruits. Where fruits are produced without fertilization of ovule. Fruits are produced without fertilization of ovule. Fine. So parthenocarpy, which is a seedless fruits, can be produced by spray of IAA, and example includes tomato. It is a selective weed killer. Why we are saying so? It is a selective weed killer. Weeds are what? Unwanted plants. So it is selective for it only kills dicots. So 2,4-D, as I mentioned, which is a herbicide or a weedicide, 2,4-D, which is a auxin itself, is used to kill the dicot weeds. It does not affect monocot plants. So when we apply 2,4-D over the plants, then dicot weeds do not have the ability to release auxin, or we can say they do not have the ability to tolerate high auxin levels, causing hormonal toxicity. It causes hormonal toxicity. So hormonal toxicity is caused in the dicots, while monocots can effectively throw out the auxin out of them, and therefore there is no hormonal toxicity in monocots. Therefore, they are the selective weed killers. Auxin can be used as selective weed killer. Example 2,4-D, which is used as a herbicide or weedicide, is an auxin that is widely used to kill the dicot weeds. This we need to remember, that it kills dicot weeds, not the monocot one. Potato dormancy. So MH, which is malic hydrazide, and NAA keep lateral buds of potato tubers dormant. So we know that this is what? Apical dominance due to the presence of auxin. So lateral buds remains dormant and the potato tubers can be stored for longer duration. So this, by preventing the growth of the lateral buds, we can prevent the potato tubers from the growth and they can be stored for longer duration for futures. So these are certain physiological effects of auxin. Now, how can we determine the concentration of auxin? So it is what actually is bioassay. So it is testing of a biological activity like growth response of a substance by employing a living material like plant or plant part. So it is testing the biological activity of auxin here, like how it is affecting the growth response by employing a living material like plant or plant part. So oxygen bioassay is quantitative test. That is, it can measure the concentration of oxygen that can produce the effect and the amount of effect. So first is the Avena curvature test. So there are two tests which are known. First is the Avena curvature test. In this, we measure curvature that is produced as a result of oxygen supply. So this experiment is based upon the experiments of Wendt in 1928. 10-degree curvature is produced by oxygen concentration. So we have created a standard where it has been shown that 10-degree curvature is produced by oxygen concentration of 150 micrograms per liter at 25 degrees centigrade and 90% relative humidity. The test can measure oxygen up to 300 picogram per liter. So we have a standard curve for that. Standard curve, which is known as the shadow graph. We have a shadow graph, which is the standard curve. Which is the standard curve. So in this, what we do is, firstly, there is a oxygen present in the tip of the seedling. So what we do is we just cut it and place it over the agar. So oxygen will diffuse into the agar. So oxygen diffuses into the agar block, and now if I put this agar block over this cut piece of the stem, then what we will see is, due to the release of the oxygen from this agar block, there will be an effect of the oxygen and it will bend. So you can notice this curvature, that how much changes in the curve is happening, and therefore based on the angle of curvature, like 10-degree curvature as I mentioned is produced by oxygen concentration of 150 microgram per liter, and conditions should also be checked whether it is at 25 degree and 90% relative humidity or at some other conditions. Depending on that, we make a standard graph and we can determine the oxygen concentration. So this is Avena curvature test, which is mainly used for the bioassay of oxygen. So this you can remember by the term curvature as well. And A for oxygen, A for Avena. So Avena curvature test, because oxygen causes the curvature. So Avena curvature test. Second is the root growth inhibition test. That how much root growth inhibition is happening. So in the test culture, you will see there is an inhibition of the growth which is treated, and in the control, root growth inhibition, root growth is being seen. Second type of plant growth regulator is cytokinin. So the term cytokinin we know was discovered by Snook and Miller, where they saw that in tissue culture there is a cytokinesis which was observed by this Snook and his co-workers. Skoog and his co-workers, where he found that when the damaged DNA was used, then cytokinesis is promoted. So cytokinesis have effects on cytokinesis, and hence the name. Cytokinesis means division of, kinesis means division of cells. Cytokinesis is the division of cells, and it was discovered as kinetin, which is a modified form of adenine or purine from the autoclaved herring sperm DNA. So when the autoclaved herring sperm DNA was used in that media, they observed the cytokinesis effect, and they found that this is because of a substance which is a kinetin, and this kinetin is a modified form of adenine or a purine. We know that DNA is made up of purine and pyrimidine. Purine and pyrimidine. Out of which guanine and adenine are purines, and thymine and cytosine sorry, and thymine and cytosine are the pyrimidines. So in the autoclaved herring sperm DNA, there is a modified form of adenine or purine, we can say, that is kinetin was discovered, and it was shown to have the cytokinesis effect. So kinetin does not occur naturally in plants. Natural substances with cytokine-like activities led to the isolation of ZRT. So kinetin is not a natural occurring cytokinin in not a natural occurring PGR in plants, but the naturally occurring cytokinin in plants is zeatin, which was first isolated from Zea mays. That is the it was first isolated from Zea mays. That is corn. So it was first isolated from corn kernels and coconut milk, and it was named as zeatin based on the substance from which it was isolated. So root tips are the major site for the synthesis of cytokinins. This is important. Also synthesized in developing shoot buds, young fruits, etc. Cytokinin plus gibberellin forms the pomelin, which is apple size and larger. And bioassays involves tobacco pith cell division test and chlorophyll prevention and retention test. So these are the bioassays for cytokinin. You need to remember these names. So for auxin, we were having the root growth inhibition test, and then we were also having the Avena curvature test. And for cytokinin, we have tobacco pith cell division. This is cell division means cytokinesis, right? So tobacco pith cell division test and chlorophyll prevention test. So in tissue culture, if low cytokinin and high auxin is provided, then it is leads to root differentiation. Root differentiation. And if high cytokinin, then we provide high cytokinin and low auxin, it leads to shoot differentiation. So this is important. Low cytokinin and high auxin, root differentiation. High cytokinin, low auxin, shoot differentiation. So more of the differentiated roots are produced and root elongation stops. Right? So principal physiological effects of cytokinin involves firstly the cell division, which is the characteristic function of the cytokinin. Characteristic function of auxin was to play a role in phototropism, that is leading to apical dominance. Characteristic function of cytokinin is cell division. It is playing an important role in cell division. Second is morphogenesis, that is changes in the morphology. Changes in the morphology. So morphogenetic changes, that is root and shoot development, are induced by cytokinin and auxin in presence of media in tissue culture. Fine. So the root and shoot development is induced by the cytokinins and auxins in the presence of media in tissue culture. Third is overcome apical dominance. So we here see that auxin and cytokinin are having an opposite effect. Such effect is known as antagonistic effect. Antagonistic, that is against each other. So apical dominance was observed in auxin. While it helps in overcoming the apical dominance, it promotes the growth of lateral buds. While the auxin was promoting the growth of the apical buds. Growth of apical buds. Both are growth promoting hormones. Fourth is delay in senescence, which is known as Richmond-Lang effect. So senescence means aging. Senescence is delayed by cytokinin as it helps in nutrient mobilization, and it also helps to produce new leaves and chloroplast in leaves. So therefore, you can see here that it is, since cytokinin is helping in production of chloroplast in leaves, therefore by chlorophyll preservation test we can check the concentration or effect of cytokinin. Fifth one is it promotes lateral shoot growth and adventitious root formation, which is opposite of apical dominance. Therefore, a balance of all the hormones is required for a well-defined growth. So these are the some physiological effects of cytokinins, which were growth promoting. So cytokinin was actually growth promoting, but its effect were antagonistic to the auxin. So if you remember the functions of auxin, you will be able to remember the physiological effect of cytokinesis. That auxin, although was helping in cell elongation and therefore was leading to cell elongation and was therefore helping in the cell division. So cell division is one of the major characteristic function of the cytokinin. But after that, if we will see, so we will see that the cytokinin and auxin have antagonistic effect in the sense that auxin promotes apical dominance while the cytokinin leads to the growth of the lateral buds. So auxin is helping in the growth of the apical buds while the cytokinin is helping in the growth of the lateral buds. Auxin is helping in abscission, in delaying the abscission, and cytokinin is helping in delaying the senescence. Next is gibberellin. So gibberellin was identified by in foolish seedling disease. Seedling disease by E. Kurosawa. E. Kurosawa, who observed the effect of a fungal infection, gibberellin, fungal infection on rice, and found it leads to the elongation. It leads to the elongation, the stem. So more than 100 types of gibberellins, that is GA1 to GA100, are known. However, GA3 is the first one to be identified. It can be represented in the form of C19H26O6, and this is the representative of all the gibberellins. So all the gibberellins in this class can be represented in the form of C19H26O6. This is important that GA3 is the representative of all the gibberellins, and they are synthesized in the buds, shoots, roots, and germinating seeds. All GAs are acidic in nature, as the name also suggests. Gibberellic, we called it as gibberellic acid. So all gibberellins are acidic in nature. First one to be identified was GA3. Representation is C19H26O6. So what are the physiological effects of gibberellin? First, we know very important characteristic function on the basis of which it was identified was stem or internode elongation. Internode is the distance between two nodes. Node from where the leaf arrives. Right? So the distance between the two nodes is known as the internode. So it helps in the stem or internode elongation, and on the basis of this only it was identified, and gibberellin induced internode elongation just prior to flowering in rosette plants. So just prior to flowering, it induced internodal elongation, example beet and cabbage. This phenomenon is known as the bolting effect. The bolting effect is due to the gibberellins. Elongation of genetically dwarf plants. So we know that Gibberellins lead to elongation. So elongation of genetically dwarf plants. So when gibberellins are applied to dwarf maize, pisum, etc., pisum is P plants, then they become tall. So therefore, it is helping in the elongation of genetically dwarf plants. Also, it is playing an important role in seed germination. How it is playing important role in seed germination? So it induces the synthesis of hydrolyzing enzymes like alpha amylase that acts on the starch, lipase that acts on the lipids, and proteases that acts on the proteins. So it induces the synthesis of hydrolyzing enzymes like alpha amylase, lipases, and proteases in the seeds, and these enzymes help in seed germination by promoting the breakdown of sperm. So endosperm, sorry. So alpha amylase that acts on the starch, which is one of the main constituent. Then lipase that acts on the lipids. So mainly lipids, starch, and proteins are present, and proteases which are present in the seeds. So mainly these three things are present, and on the basis of that, it promotes the breakdown of endosperm. Increase the length of grape stalk as well as elongate and improve the apple fruits. We know that larging of apple. So we have seen that it increases the length of the grape stalks as well as elongated and improve the apple fruit. So these are the physiological effects of gibberellin. You need to remember these effects. Firstly, elongation is the very important and is a characteristic function of gibberellin, where it is promoting the stem or internode elongation. Then elongation of the genetically dwarf plants. So genetically dwarf plants like maize, spice, etc., can become tall under the influence of the gibberellin. So when gibberellins are sprayed over them, then they elongate. Third is the seed germination. So it is inducing the synthesis of hydrolyzing enzymes like alpha amylase, lipase, and proteases that helps in the synthesis of the enzymes that finally results in the seed germination by promoting the breakdown of endosperm, and it leads the increase in the length of the grape stalk. Also, GS speed up malt formation. So it is helping in the malting process to help in growing wine industry. This is commercial use. Increase height of sugarcane plant. We know that it can act on the nodes and internodes. Therefore, it can increase the yield of sugarcane up to 20 tons per acre. GA delays the senescence in plants, that is, delayed the aging. So the fruits can be left on the tree longer so as to extend the market period by application of the GA. And the next is bring juvenile conifers with GA, hastens the maturity period, thus leading to early seed production. So maturity in conifers is induced by GA. Therefore, rapid seed production. Right? So these are the three growth promoting hormones. The next is abscisic acid, which is a growth inhibiting hormone. So abscisic acid is a growth inhibiting hormone, and it was discovered for its role in regulating the abscission and dormancy, that is the period of suspended growth. Suspended growth. But like other PGRs, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor. So it is a plant growth inhibitor and an inhibitor of plant metabolism, resulting in dormancy. Is mainly synthesized in senescent organs only. So it is usually a stress hormone. ABA is also known as a stress hormone. It is known as the stress hormone. So you can remember that only mainly old people have a lot of stress. So senescent organs and old leaves are the sites of synthesis of ABA. It's a stress hormone. It is also known as stress hormone because it protects the plant from adverse condition by like water stress by inducing the closing of stomata. So it is actually preventing the plant from stress condition. Therefore, it is also known as stress hormone. So, in case of conditions like water stress, adverse conditions of water stress, it induces the closing of stomata, thus preventing the loss of water from the plant. ABA increases tolerance of plants to various types of stresses. As I mentioned, it is a stress hormone. It increases tolerance of plants to various type of stresses. Firstly, what are the physiological effects? As I mentioned, it induces senescence and abscission. So this is a growth inhibiting, right? And it mainly acts on the old parts of the plant. So old parts means it induce senescence. So ABA causes senescence and abscission of leaves and fruits by increasing the activity of cellulase and pectinase enzymes. It induces bud and seed dormancy. We know it affects the metabolism. So it increases the bud and seed dormancy. It inhibits seed germination. Unlike GA that was helping in the seed germination, it is inhibiting the seed germination. ABA plays a major role in seed development, maturation, and dormancy. And by inducing dormancy, it helps seed to withstand the desiccation. So it is also a kind of stress mechanism, right? The dormancy of the seed is also a kind of stress mechanism. Therefore, ABA is actually helping the seed to withstand the desiccation and other factors which are unfavorable for growth by inducing the dormancy. Third is stomatal closing, as I mentioned during the water stress. So ABA closes stomata under the water stress condition, anti-transparent. Transpiration is the process of evaporation of water from the stomata of the leaves, through the stomata of the leaves, right? Through the stomata. Just pulling up of water from the root to the leaves and the evaporation of water. In most situations, it acts as antagonist of GA. Therefore, it is also known as anti-GA. So, as I mentioned, like for example, GA that was actually promoting the seed germination. Abscisic acid is inhibiting the seed germination. Therefore, it acts as an antagonist. Antagonist means against GA. Opposite effect. The next one is ethylene. We know that ethylene is one of the gaseous. It is the gaseous plant growth regulator. It is the gaseous plant growth regulator which was discovered firstly by H. Cousin. So ethylene can be included in both the groups as I mentioned. It can sometimes act as a growth promoter as well as growth inhibitor. But mainly it is a growth inhibitor hormone. So if the option does not provide option, like if it has provide growth promoter, growth inhibitor, option C is both A and B, option D is none A and B, then both A and B is fine. But if both option is not given, then growth inhibitor is the right one. So ethylene is a simple gaseous PGR. It is a hydrocarbon. So we know it is a hydrocarbon. So it is a hydrocarbon. This is ethylene. and it is helping in the fruit ripening. This is very important. You might have seen that if we keep some fruits with the ripe bananas, the fruit also become ripened very fast. So this is a gaseous molecule which is released by the ripened fruits and helps in the ripening of the unripe fruits. So it is also known as ripening hormone, and it is one of the most widely used PGR in agriculture for the early ripening of the fruits. Biosynthesis of ethylene takes place by methionine amino acid. Ethylene is synthesized in large quantity by ripening fruits and senescent organs. What is the physiological effect? Firstly, post-harvest ripening of fruits. So banana, apple, tomato, etc. Nowadays, ethipone or CEPA, that is chloroethyl phosphonic acid, is used at commercial level as a source of ethylene. So these terms are important. Ethipone or CEPA, that is chloroethyl phosphonic acid, is used at commercial level as a source of ethylene, and it enhances the respiration rate during the ripening of the fruits. This rise in the respiration is called the respiratory climatic. This is important. Second is the stimulation of senescence and abscission of flowers and fruits. So thinning of cotton, cherry, walnut, etc. because of the stimulated senescence and abscission of the flowers and the fruits, because it is stimulating that. We know it is a plant growth inhibitor, right? So therefore, it will stimulate the senescence and abscission. Third is flowering and synchronizing fruiting in pineapple. Flowering in mango is being affected by the ethylene. It has a triple response on stem. This is important. Apical hook formation in dicot seedlings, swelling of the axis, and horizontal growth of the seedling. So this is known as the triple response on the stem, which is due to the ethylene. So it leads to the apical hook formation in dicot seedling. It leads to the swelling of axis, and also it is leading to the horizontal growth on seedling. So what is apical hook? It is an exaggeration exaggeration of apical hook's curvature. It promotes that, it exaggerates that apical hook curvature. So apical hook formation, so it leads to the apical hook formation in dicot seedling. It leads to the thickened hypocotyle horizontal growth of fruit. Therefore, it leads to inhibition of hypocotyle and root cell elongation. And also, it leads to the radial expansion, that is swelling, radial expansion or horizontal growth of the seedling by performing the radial expansion of cells in hypocotyle. So these are the physiological effects of the ethylene. Next is promotes root growth. As I mentioned, ethylene promotes root growth and also stimulates the formation of root hairs, thus helping the plant to increase their absorption surface. Femaleness, it induces feminism effect in cucumbers, promotes female flowers to increase the production. Seventh is ethylene breaks seed and bud dormancy. It initiates germination in peanut seeds. That is why we said that it has both promoting as well as inhibiting effect, right? Because in certain cases, it is leading to the promotion of the by initiating the germination in peanut seeds, sprouting to potato tubers, etc. Fifth, last effect of ethylene is it promotes rapid internode petiole elongation in deep water rice plants. It helps leaves, upper parts of the shoot to remain above the water. So rapid internode or petiole elongation in deep water rice plants so that the shoot can remain above the water. So this is the summary of different plant hormones that we discussed today, that where they are produced and what are the major functions. So I will quickly go through it. The first one, which is the auxin or the indole acetic acid. The first one is the auxin or the indole acetic acid. So it is present in the root, apical meristems, and young leaves are the primary sites. Shoot apical meristems. Root apical meristems, as I mentioned, also produce auxin, although it depends on the shoot for much of its auxin. Developing seeds and fruits contain high levels of auxin, but it is unclear whether it is newly synthesized or transported. Major function is it stimulates stem elongation at low concentration only, promotes the formation of lateral and adventitious roots, regulates development of fruit, enhances apical dominance, functions in phototropism and that is gravitropism, means against the gravity. Promotes vascular differentiation. We know xylem differentiation retards leaf abscission. The next is cytokinin. So cytokinins, these are synthesized primarily in roots and transported to other organs. Although there are many sites of production, it regulates cell division, the major function in shoots and roots, modify apical dominance, and promotes lateral bud growth. It promotes movement, that is nutrient mobilization, and stimulates seed germination and delay the leaf senescence. Gibberellins, also known as gibberellic acids, which are acidic in nature, they are mainly found in the meristem of apical buds and roots, and they are the primary sites of young leaves are the primary sites of production. It stimulates stem elongation. This is the major characteristic function of gibberellins. And next is brasinosteroids. So brasinosteroids are the recently discovered. These are present in all the plant tissues. They're although different intermediate predominant in different organs, internally produced brasinosteroids act near the site of synthesis. So they usually act locally. It promotes cell expansion and cell division in shoots, promote root growth at low concentration, inhibit root growth at high concentration. So this is important, that at low concentration of this, root growth is promoted. At higher concentration, root growth is inhibited. It promotes xylem differentiation and inhibits phloem differentiation, promotes seed germination and pollen tube elongation. So you must remember the function of brasinosteroids. Next is abscisic acid. We know that they are the stress hormones usually present in the old senescent tissues, and it inhibits their growth, promotes stomatal closing during the drought stress. It inhibits the growth, promotes seed dormancy, etc. Next is strigolactones. So strigolactones are the recently discovered plant growth regulators. These carotenoid-derived hormones and extracellular signals are produced in roots in response to low phosphate conditions or high auxin flow. So it promotes seed germination, control of apical dominance, and the attraction of mycorrhizal fungi to the root. So therefore, this is important. And the last one is ethylene, which is a gaseous hormone produced by most of the parts, and if it is produced in high concentration by the senescent and during the senescence and during the abscission and the ripening of some types of fruits. Synthesis is also stimulated by wounding and stress. It promotes the ripening. This is very important characteristic of the ethylene, that it promotes ripening of fruits, and the triple response, that is inhibition of stem elongation. So elongation, that is increase in the height, inhibits, and it increases laterally. So lateral expansion and horizontal growth is promoted. Enhances the rate of senescence, promotes root and root hair formation, promotes flowering in the pineapple family. We know it causes femaleness. That is, feminism character is induced. Feminism is induced, right? So these are the characteristics of ethylene. So these are the major plant growth regulators, and with this, these are the plant growth regulators that we see play important role in the growth as well as in the development. So thank you for attending to